When was the last time you had to go to the bathroom? I'm guessing probably not pretty long ago, right? And you have your kidneys, at least in part, to thank for that. Specifically, your renal tubules are the part of your kidneys that filter waste and toxins out of the bloodstream into your kidneys to make urine. And it would be pretty bad if something were to happen to such important organs, right? Like cancer, for example. Cancer is a disease caused when mitosis occurs at a rapid rate, almost unregulated, and your cells will start growing at an exponential rate and spread to all the organs in your body through a process called metastasis, where the cancer cells use your bloodstream as a means to reach the other organs in your body. Now, because cancer spreads to all of the organs in your body, it can permanently disrupt the functions of each of your organs and throw your entire body into jeopardy. Specific to your kidneys though, clear cell renal cell carcinoma, or CCRCC, is the most lethal type of renal cancer found in adults today. That's crazy. It kills over 85% of all renal cancer patients. How can we use things like genetic engineering to help solve this huge problem? Today, I'm going to be talking about a gene therapy I designed to do just that. It uses gene silencing of a gene found in clear cell renal cell carcinoma to help slow down or even stop the metastasis of CCRCC. Stay tuned to find out more. Just last year, research was done by a group of Chinese scientists to identify the main genes involved in the metastasis of CCRCC. One of them is called cyclin-dependent kinase inhibitor 3, or CDKN3. And that will be the target gene for this gene therapy. I've already talked a bit about the main mechanism of this gene therapy, which is gene silencing. But what exactly does that mean? So imagine you have a switch that can turn all of your genes on and off. RNA interference is the process of switching a light switch on and off. And the RNA interference construct is that switch. RNA interference is the process of using single-stranded RNA, or siRNA, to bind to a gene and cut it up so that its function is completely disrupted, literally turning its function off. If this can be applied in a long-term manner, which it can, to cancer cells, we can potentially stop cancer in its tracks. In order for the gene therapy to reach its target cells, it needs a vector to get it there. And so the vector for this gene therapy is a viral vector, specifically the Newcastle disease virus, which is a perimyoxyvirus, which is found in birds and causes things like conjunctivitis in humans. But this experiment uses a non-pathogenic strain, so hopefully no kidney cancer patients will get conjunctivitis as a byproduct. Newcastle disease virus was chosen specifically for this therapy because it's been shown as a promising oncolytic virotherapy. Oftentimes what happens as a viral vector travels through the bloodstream to its target tumor cells is that it gets lost due to the sheer amount of soluble factors and proteins that exist in the bloodstream. So what we have to do is attach a tumor-specific ligand to the Newcastle disease viral vector, or the NDV viral vector. The tumor-specific ligand for this experiment is a combination of a gene carrier known as PEA and a cytotoxic agent known as LTVQVG-RVH. This cytotoxic agent is an amide group that is connected to the PEA gene carrier through an amide bond formed between the amine group of the PEA and the carboxyl group of the cytotoxic agent. Ideally, this is what would happen. The NDV vector would inject the siRNA specifically to the target cells, thanks to the cytotoxic agent. Ideally, this is what would happen. The NDV vector specifically is able to deliver the siRNA thanks to the cytotoxic agent. The siRNA, once injected, will then bind with something known as the AGO enzyme to form the RISC complex. Because the siRNA is complementary to CDKN3, it will bind the whole complex to the transcribed mRNA of the CDKN3 gene, and then the AGO enzyme will chop it up, thus disrupting the function of the CDKN3 gene. Thank you so much for watching. I hope you enjoyed hearing about the cancer gene therapy I developed. If you or anyone you know is a cancer researcher who has a lab and is willing to help me develop my passion project, then please reach out to me using the link to my email listed down below, as well as the link to my LinkedIn profile if you'd like to further connect with me about genetic engineering and its applications. Until next time.